So now we're going to look at Chapter 19, Blood, in sort of a general view. We've already looked a little bit at the function of red blood cells. We've looked at ABO blood typing and RH. We've looked at some of the white blood cells. We've looked at the 5 types of white blood cells and what they do. Now let's just take a more general view of blood. So first we're going to talk about the major functions of blood and the components of blood. So the components can be broken down into the formed elements, which are things like red blood cells, white blood cells, and platelets, versus the plasma. And as we recall, blood is a fluid connective tissue, and it is basically going to contain proteins that are dissolved in what we could consider a ground substance, and in this case those proteins are globular proteins that are in a fluid ground substance, and this combination we call the plasma. So we have a specialized matrix of blood called plasma, and then we're going to have our formed elements, which are our blood cells and our platelets, that are going to be a suspension within this plasma. We'll look at the red blood cells and how they are specialized oxygen carrying machines, and then how they have a particular shape that allows them to maximize their surface to volume ratio for very rapid exchange of dissolved gases. We will see that these cells have lost their cellular organelles. They have no nucleus, and most of their cellular organelles have long since been ejected so that they can pack themselves full of a molecule called hemoglobin, which is specialized for carrying oxygen. We'll talk a little bit about the way the body makes blood cells, both red and white blood cells. Red blood cells are made in a process called erythropoiesis, which is just basically making red blood cells. We'll also talk about leukopoiesis when we talk about white blood cells, and hematopoiesis or hemopoiesis is the process of making the formed elements of blood. We'll also talk about the blood typing. Once again, we'll review that, and we'll talk about the different types of antigens we can see displayed on a red blood cell, and this can be A, B, and RH. And we'll talk about how the combinations of these antigens can give rise to the ABO plus and minus blood types that we see when we look at the major blood types. Then, of course, we can revisit our white blood cells, our 5 white blood cell types, and we can look at some of the things that influence their production. We can talk about the platelets, and the platelets are actually really related to the next item on this list, which is hemostasis, which is the stopping of the flow of blood. So platelets are one of a number of things, mechanisms that blood has to prevent blood loss in the event of an injury to the blood vessel wall. So if you cut the blood vessel wall and you create a means of egress of blood from that blood vessel, then you can disturb homeostasis because you could disturb the fluid balance in the body, and you could also diminish the body's capability of carrying oxygen and nutrients to the tissues and removing waste. So these are the major important outcomes that we have from this chapter. Now, one of the things we know is blood is part of the cardiovascular system, and the cardiovascular system is comprised of the pump, which is the heart, which is providing the mechanical force to move blood through the series of conducting tissues, the blood vessels, that carry this blood, the fluid connective tissue, all throughout the body. And as we said, blood is a specialized connective tissue, and the cells, the formed elements, are suspended in the fluid matrix that we call plasma. So just as a reminder, plasma is the special term we give to the matrix, the fluid matrix of the blood connective tissue. When we look at the functions of blood, one of the main things that we think of first is the transportation of oxygen to the tissues and carbon dioxide away from the tissues, as well as nutrients such as glucose, and taking away things like lactic acid, bringing things like hormones to the cells, to the target tissues, and removing metabolic waste, as we've said, like lactic acid. Another thing blood is important for is regulating pH. We'll see we have a blood buffer system, and we'll also see that the ionic composition of blood is going to be very important, and changes to the ionic composition of blood in terms of calcium ion, sodium ion, potassium ion, other ions, is going to affect the interstitial fluids and the levels of interstitial fluids and the exchange of fluid between the blood and the interstitial fluid. And later on, we'll learn about fluid compartments in the body and how the fluid in the fluid compartments of the body has to remain balanced in order for the organism to maintain homeostasis. So we're going to see how blood composition is going to affect the fluid components and the fluid compartments of the body. And we'll see how blood has within it certain factors, including platelets. Platelets are actual formed elements, but it's going to have factors such as fibrinogen and calcium and other things that are going to be very important for initiating the hemostasis or the blood clotting cascade so that we can prevent excess fluid loss during a traumatic event or some kind of injury to the body, especially one that dissects blood vessels or just creates an opening so that blood could leave. We're also going to see that this is going to be the mechanism or the transportation system for our white blood cells. And our white blood cells are important in fighting toxins and pathogens. And as we will see, oftentimes these white blood cells will start off circulating in blood, and then they'll migrate into tissues where they will do their work to engulf pathogens or to neutralize pathogens. As we've seen in previous chapters, blood is important for stabilizing body temperature as well, and we've seen this in the sweating mechanism, so that if the body temperature rises, then blood vessels near the surface of the skin will dilate and the sweat glands will activate, and then we can radiate heat away from the body by losing vapor, water vapor, and we can cool the blood this way, and we can carry the heat out of the blood into the atmosphere by basically releasing the sweat. And this water vapor will carry heat away with it. By the reverse token, if somebody is excessively cold, then blood vessel dilation stops and blood vessel constriction occurs in the skin, so that we are now shunting blood away from the surface and into the inner core of the body. And the other thing we'll start to see is muscles will start to shiver, and this creates heat. When we look at blood, we will see that it has a slightly higher temperature than body temperature measured under the tongue, typically. It has a fairly high viscosity for a fluid medium. So unlike other connective tissues, which are not fluid, blood is actually pretty fluid. I mean, it is a fluid. But it has a much higher viscosity, that is a thickness, than, say, pure water or other substances like water. So we'll see that because of the formed elements that are in blood, it tends to flow less quickly, and it creates more of what we call peripheral resistance. So it has a resistance to flow due to the high viscosity. We'll see that it's slightly alkaline. So the typical homeostatic window of blood alkalinity is, or I should say a blood pH in general, is between 7.35 and 7.45. So if we see something where blood dips below 7.35, then we have a condition called acidosis. If it rises above 7.45, we have alkalosis. And mild acidosis and mild alkalosis can be usually corrected for pretty quickly by compensatory mechanisms of the body. If, on the other hand, we get too far outside of these windows, then we could have a serious problem that could result in death if not dealt with fairly early on. When we look at blood volume, we'll find that it's about 7% of body weight. Just as a reminder, the weight of 1 liter of water is 1 kilogram, so approximately 2.2 pounds. Because of the formed elements in blood, blood's going to weigh a little bit more. But if we take a 75 kilogram person, this is sort of an average weight, that person would have approximately 5.25 liters of blood. When we look at whole blood, we can break it down into its component parts, the fluid component, the plasma, and that's usually the straw-colored stuff. And if you spin it down in a centrifuge, what you'll find is you'll get the formed elements at the bottom and the plasma at the top. So the bottom half is usually red blood cells. and it looks kind of a dark red. And then you have this tiny little buffy coat of white that are the white blood cells sitting on top of the red blood cells. And then on top of that, you see the plasma, which is the straw-colored fluid. And so that is this process of fractionation, is when you separate the whole blood into the formed elements and the plasma. And the formed elements settle on the bottom when you put them in the centrifuge. So when we look at plasma, it's going to make up about 55% of the entire blood volume, and 90% of that plasma is water. Now, the other thing that plasma has in it is proteins, and it has dissolved proteins called albumins and globulins. And these proteins are actually, as the name implies, globular, so they're round. Unlike in other connective tissues where we see a lot of fibrous, insoluble proteins, these proteins are soluble and they're globular. We'll also see enzymes and other things in plasma as well. So we'll see that the plasma is going to have some similarity in composition to the interstitial fluid, that is the fluid between the cells. And we'll see that this is going to be important when we look at the transport of things across the capillary wall, such as nutrients and dissolved gases. Now here is a vial of blood, and this looks like whole blood. If we were to spin this down and fractionate it, we'd get about 45% of blood would be formed elements, and then the top 55% could be plasma, and there could be some variation in that. Our formed elements, of course, consist of our red blood cells. That's about 99% of our formed elements. Our white blood cells are few and far between. You will see an elevation in white blood cells if there's an infection or something like that, but typically they are less than 1/10 of 1%. And the same is true for platelets. They are less than 1/10 of 1%. And that is why typically when you fractionate blood and you get what's called the hematocrit, that's the formed elements of blood, you'll see the majority of that is made up of red blood cells, or what we call erythrocytes. Now the plasma, as we know, is 90%, a little bit more than 90% water. Then we've got our plasma proteins, and then we have ions and other solutes. When we look at our plasma proteins, we'll see that we've got albumins and globulins. And both of these can function as transporters, so they can transport substances through the blood. And sometimes we see the globulins include the antibodies called the immunoglobulins, and we'll study those when we get to the lymphatic system and immunity. We will see the transport globulins and the albumins that transport substances are very important, because a lot of times these things could not be transported through blood without them. We will see especially the metalloproteins are very important, because these will grab free metals that are floating around that you don't want floating around in blood. So for example, iron, zinc, magnesium, these kinds of things. So, well, iron and zinc and copper are good examples. So you don't want those things floating around and reacting with things. So you really have these metalloproteins that are going to hang on to things like iron, zinc, and copper, because these things can react with other things in the body in a way that you don't want them to. So you really don't want certain free metals just running around in the body. We also have, of course, our steroid binding proteins, and these are going to be escorting our steroid or our cholesterol derivative hormones around through the blood. Now, we also talked about fibrinogen a little bit earlier. I mentioned that, and that is a globular protein that is made in the liver. And when the body has to initiate a clotting cascade, or the blood will initiate a clotting cascade, this fibrinogen, which is this globular protein, will be converted into something called fibrin. And fibrin is a fibrous protein, as the name implies. It will make these networks, or this meshwork, that acts as a net, and it will plug up the area where the injury has happened. It will trap other formed elements like erythrocytes, platelets, and so forth, and it will stop the blood flow through that cut, or that means of egress that shouldn't otherwise be there. And then, of course, we've got our other plasma proteins that consist of varying things. Now, when we look at where the plasma proteins come from, most of them come from the liver. We'll see that our immunoglobulins will be produced by a certain type of cell called plasma cells. And these plasma cells are mature versions of B lymphocytes. And we'll study lymphocytes a little bit more, especially when we get to lymphatics and immunity. But we have several types of lymphocytes, one of which produces antibodies, and these are B lymphocytes that mature in bone marrow and produce antibodies. So it's really easy to remember that B and antibodies. Eventually the B lymphocytes, when activated, will convert into plasma cells and start churning out antibodies. We also have peptide hormones that are made by endocrine organs. These will also be circulating in blood. Now, when we look at our formed elements, once again, we've got our red blood cells. These are called erythrocytes. We've got white blood cells, and we got 5 types of them, which we'll review again in a little bit. And these are called leukocytes. And then we have these cell fragments called platelets. And platelets are these little tiny things that are membrane-bound packets of cytoplasm with certain enzymes and things in them to promote blood clotting. And these are much, much smaller than either of the cell types. Now, in humans, platelets are not cells. In other animals, they have something called thrombocytes, which are cells, and they serve the same function as platelets. So sometimes you hear the word thrombocytopenia, which is a dearth or lack of platelets. And in humans, it's a little bit of a misnomer because platelets are not thrombocytes. Thrombocytes are actual cells, and we do see thrombocytes in other animals, but in humans, platelets are not cells, and they don't have any cellular organelles. They're just little membrane-bound packets. Now, when we talk about the production of any of these formed elements, we call this hemopoiesis, or sometimes you'll see this hematopoiesis. But either way, both terms are apropos. They are used to describe the production of any one of these types of formed elements, or all of these formed elements. And when we talk about each of the formed elements separately, for example, red blood cell production is called erythropoiesis. White blood cell production is called leukopoiesis. And then we can have thrombopoiesis as well, which is the production of platelets. When we look at platelets, they're little teeny tiny things, and we've seen them under the microscope before. They are these little purple spots in a blood smear slide on a microscope, and they're basically just cytoplasm surrounded by a membrane that have special enzymes and clotting substances in them, so that when they are activated, they will start forming these little platelet plugs. And we'll visit that later on when we look at the process of hemostasis, or stopping of blood flow. Then we've got our 5 types of white blood cells. As you remember, we have got our neutrophils, our lymphocytes, our monocytes, our eosinophils, and our basophils. And notice that I went through them in the order that they appear in circulation, so by percentage. So we have the highest percentage of neutrophils in circulation, followed by, well, if we follow our acronym, never let monkeys eat bananas. So that's the way to remember the order of white blood cell, or the percentage of white blood cells in circulation. We've got our neutrophils, which are first responders. They're going to get to the site of injury first, and these are going to be one of our macrophages. They will engulf things. And they are also one of our granulocytes, in that they have these little granules in them that are quite visible. In fact, if we look at our neutrophils, our eosinophils, and our basophils, these are our granulocytes. These two, the lymphocytes and the monocytes, are our agranulocytes. And as we recall, neutrophils are our first responders. Eosinophils are really good against parasites and flukes. They also are involved in allergic reactions. Basophils are very important for beginning an inflammatory cascade. They have heparin and histamine in them. So we have the ability to start inflammation as well as increase blood flow, because heparin is a blood thinner. And we look at lymphocytes. These are going to be part of our acquired immunity. So these cells are going to be specific to certain types of pathogens. So we've got really 3 types of lymphocytes, 2 of which are specific to pathogens, to a specific pathogen, and they will develop an immune response to a particular pathogen. So we've got T lymphocytes, which develop in the thymus, hence the name T lymphocytes, and these will be important for cell-to-cell mediated immunity. We have B lymphocytes that develop in bone marrow, hence the name B lymphocytes, and they're the ones that will, when activated, convert into plasma cells and start producing antibodies. We also have something called natural killer cells. These are a type of lymphocyte that will go around looking for any abnormal cell. Maybe it's cancerous, or maybe it has mutated, or maybe it has been infected. And it's not too particular about what the pathogen is, but it will also kill that cell in a cell-to-cell mediated response. We've got our monocytes over here, which, as you may recall, are our monsters. They're very large. They migrate from the blood to tissues where they mature into macrophages. So these are macrophages. So we've got 3 types of cells here that are phagocytic. And our macrophages are one of them, our monocytes. The other 2 are the neutrophils and the eosinophils. So these are also phagocytic. These are called microphages. We also have another distinction in white blood cells that we should remember, are granulocytes. And the granulocytes have little granules in them. In fact, these do too. You just can't see them as easily because they don't have the affinity for dyes that these do. But in any case, these are called granulocytes, and they're immediately recognizable because they have stained granules in them that you can see. And they all end in fils. So you can think of fils as they're filled with something. So the granulocytes are filled with granules, substances in their granules that stain a color that makes them show up more on microscope slides. Over here in our agranulocytes, you don't see the little granules anywhere nearly as clearly. So monocytes have granules as well. And the monocytes are immediately recognizable not only by their size, but by this big kidney bean-shaped nucleus. When we look over here at these 3, the neutrophils, eosinophils, and basophils, they all have something called a polymorphonuclear nucleus that has multiple lobes and has kind of a funky shape to it. Now the basophils, you can almost never see the nucleus because the granules stain as dark as the nucleus. So usually the nucleus is somewhat hidden. The lymphocytes, when you look at them on a slide, have a large nucleus with a little bit of cytoplasm around the outside. Sometimes they look like a big eye looking at you. So those are our white blood cells. Our red blood cells, as we can see, these are our oxygen-carrying machines. They have ejected their cellular organelles and their nuclei, and they are basically just what we call a biconcave disc. And they have this sort of pink donut appearance or purple donut appearance if you look at them under a microscope slide, because they're thicker on the outside and thinner on the inside. And if you look at them in cross-section, here's one that's on its side. If you were to slice through it, you would see this figure 8 where the outside is thick and then it thins in the middle and gets thicker again on the outside. And this is going to maximize the surface to volume ratio. And this is going to allow these things to exchange materials across them very, very quickly. These cells have no nucleus. They're called anucleate because of that. And they also don't have any mitochondria, for obvious reasons, because if there were mitochondria in here, they would start sucking up oxygen to use for cellular metabolism. Now these cells have no mitochondria because their job is to carry the oxygen to peripheral tissues, not to use it up themselves. So any kind of metabolism that goes on in the cell is going to be anaerobic. Let's go the other way now. So these are called erythrocytes, and as we know, these red blood cells make up 99.9% of the formed elements. We'll look at the structure of hemoglobin in a little bit. This is the protein that these cells are packed full of, and it is a 4-subunit protein that each subunit contains one heme group, and the heme group is what's responsible for transporting the oxygen. When we look at red blood cell counts, and we look at the number of red blood cells per microliter of whole blood, we see that males have anywhere between 4.5 and 6.3 million, females 4.2 to 5.5 million. One of these differences owes a little bit to the size difference. And also, the adult female is not going to make quite as much red blood cells as the males do because females have a little bit less testosterone, obviously, so they're going to, interestingly, it turns out that testosterone promotes the production of erythrocytes or red blood cells, whereas estrogen does not. When we look at the hematocrit and that is the packed cell volume, or the percent of formed elements in blood, we see that the percent of formed elements, and this might be a little bit better measure than this, is about 46% in males and 42% in females. So a slight difference there. Again, once again, due to the fact that testosterone promotes the production of red blood cells. When we look at the structure of red blood cells, we see they are, once again, highly specialized for carrying dissolved gases, especially oxygen. And they are these what we call biconcave discs. That just simply means that if we look at them in cross-section, they look like a figure 8. Here we see them in the microscope slide, our little pink donuts with our thin center. It's not hollow, it's just thin. And we can see a few platelets in this particular slide. And this is just simply under a light microscope. If we look under a scanning electron microscope, we see something that looks like this, false colored of course, but you can see the shape, how these are thick on the outside and thin in the center. They'll also stack together, somewhat like Pringles potato chips, into what we call rouleau, which is a French word that just basically means a stack of these things. And this allows them to squeeze into spaces that they otherwise would not be able to get into. So some of the capillaries that these things flow through are actually thinner in diameter than the red blood cells themselves. So they are quite bendy, and they can bend owing to the fact that their center is thinner. And so the thicker part can kind of bend, and they can squeeze through these capillaries. The other thing that's important to know is if they stack up in these rouleaux like this, then they can proceed in an orderly manner. Whereas if they just went in there all mumbo jumbo, they would create a log jam and nothing would get through. So this is another reason for the shape. Not only is it optimized for carrying oxygen and dissolved gases and exchanging gases across the surface, but it's also optimized for being able to move in very small spaces. So if you take a cross-section of one of these things, you see it's thick on the outside, thinner in the center, and it can range anywhere between 7.2 and 8.4 micrometers, and that varies, of course, for red blood cells. But one other thing that's important to note, once again, it has no cellular organelles. There's no nucleus. That's the first obvious thing about it. There's no nucleus. These things can't reproduce themselves. They can't repair themselves. They've got no ribosomes, no endoplasmic reticulum. They just carry oxygen. So they tend to wear out, and they'll wear out, and about every 120 days they have to be replaced. And they're constantly being put into circulation and constantly being broken down. So one of the things we've already talked about is their shape allows them to increase their surface to volume ratio. They can form these stacks called rouleaux, and they can bend when entering small capillaries. Here is a picture of some of these rouleaux. You can see how they stack up, and they can get through these very thin diameter capillaries. Whereas if you just had a bunch of them just moving in there, they might create a log jam if they didn't create these nice little rouleaux. Now we can also remind ourselves they lack nuclei, they have no mitochondria, as we've said. This is important because we don't want the mitochondria stealing the oxygen that the blood vessel, the blood cell is trying to carry. Has no ribosomes, they don't divide, they don't make proteins, they don't repair damage. That is why they only live about 120 days. Now when we look at the hemoglobin is the protein that carries the oxygen. And we will see that we've got 4 subunits of this hemoglobin. Each one of these subunits of this quaternary structure of protein is going to have one heme group in it, so each hemoglobin can carry up to 4 oxygen molecules. And you can look at the difference in hemoglobin between males and females. It doesn't differ that much, but we have about 14 to 18 grams per deciliter of whole blood in males and 12 to 16 in females. So not that big of a difference. This is going to vary greatly with a number of factors. So where you live, if you live at higher altitude, might be higher than if you live at lower altitude. If you are an endurance athlete or a distance athlete, you might have more. So there are any number of factors, including your diet. If you have a diet that does not have enough iron in it or enough B vitamins in it, you may actually have less of this hemoglobin, and you may have fewer red blood cells as well. So there are factors that will make this vary. When we look at the hemoglobin complex itself, we see that it's got 4 independent proteins whose tertiary structures, we've got two that are the same. We've got an alpha chain, we've got two of these alpha chains and two of these beta chains. So remember, each one of these individual proteins has a complex tertiary structure of its own. In the middle of it is a heme group, and the heme group is based on iron. So this is an inorganic molecule with iron at the center of it. And as we know, iron likes oxygen. This is why things rust in the presence of oxygen. So iron has a high affinity for oxygen. These heme groups have a high affinity for oxygen. But as we will see and study in a great deal of detail in the respiratory section, the affinity of each of these heme groups will change as the conformation of the protein changes. So as we bind one oxygen to a heme group, that's going to make the other heme groups have a higher affinity for oxygen, and they'll bind oxygen as well. And this will be important as this hemoglobin molecule passes from areas of high concentration, or I should say low concentration, to high concentration. So it'll pick up oxygen where it needs to and let go of oxygen where it needs to. And we'll talk about that in a lot of detail when we get to the respiratory system. But suffice it to say, you have this quaternary structure of hemoglobin that consists of two beta chains and two alpha chains that are identical to one another. So the alpha chains are identical, the beta chains are identical. And in between, or at the center of each of these independent proteins, is this heme group. So we've got 4 heme groups per hemoglobin. When we look at fetal hemoglobin, it's a little bit different. And what's going to happen is with the fetal hemoglobin, it's going to have higher affinity for oxygen. It's going to bind oxygen more readily than adult hemoglobin because basically it's competing with the mother's blood for oxygen. And of course, it's going to have to take up oxygen from the maternal blood at the placenta, so it has to have a higher affinity for that blood, for that oxygen, so that it will pull it away from the maternal blood. So this is an important point to make, that fetal hemoglobin is a little bit different from the hemoglobin of the mature adult. Now, when we look at the hemoglobin itself, it contains, or I should say, red blood cell itself. It's going to contain about 280 million hemoglobin molecules. Each of those can bind 4 oxygen molecules. So overall, we're going to be able to carry over 1 billion molecules of oxygen in one red blood cell. That's quite a bit. And we're going to see that the way the exchange of gases works is they follow their concentration gradients. And we will see that oxygen will move from an area of high concentration to low concentration, so from the blood to the peripheral tissues, which are using up that oxygen in their cellular metabolism, creating a dearth of oxygen, and they will be producing carbon dioxide, which is going to increase the carbon dioxide in the tissues, whereas the circulating blood in the capillaries, as it's coming through the tissues, is going to have low carbon dioxide. Then we're going to see the exchange of these materials. So oxygen goes one way, carbon dioxide goes the other way. So the red blood cells will pick up the carbon dioxide and carry it into venous circulation. Now, one of the things that's important to note that we'll get to a lot more detail in the respiratory section is while red blood cells love to carry oxygen, they don't have as much an affinity for carbon dioxide. We'll see that we have 3 ways of transporting carbon dioxide in blood, and one of the major ways is dissolved as carbonic acid. So we'll look at that in more detail a little bit later. But when carbon dioxide does bind to hemoglobin, we will find that it does not bind to the heme group. Instead, it binds one of the amino terminuses, or termini. It binds the amino termini of the hemoglobin. Hence, we get something called carbaminohemoglobin. And we'll see this is one way of 3, that carbon dioxide is transported. Now, once we get to the lungs, we have this deoxygenated blood that has made the trip through what we call the peripheral circuit to take blood with oxygen in it to the tissues. It's dumped the oxygen off and picked up the carbon dioxide from the tissues, where it goes back to the heart via venous circulation. The heart will then pump it to the pulmonary circuit, which pumps that blood to the lungs, and now it's going to release that carbon dioxide into the lungs to be off-gassed, and it's going to pick up oxygen from the lungs where you've just taken in oxygen from the air. So one of the problems is if you don't have enough red blood cells or hemoglobin, you end up with anemia. And so sometimes anemia can be associated with low iron content in the diet, but it can also be associated with low B vitamins. And so there's a particular type of anemia called pernicious anemia, and this occurs when you have low levels of vitamin B12. So vitamin B12 is very important for proper blood cell development, erythropoiesis, and so forth. So we got to be careful in our diets to make sure that we get enough vitamin B12 and we get enough iron. Otherwise, we really can't have proper red blood cell formation. As we've said, red blood cells don't last that long. So we're constantly making new ones in bone marrow, red bone marrow. And it turns out that we're making about 3 million new red blood cells that are going into circulation per second. That's quite a bit. So the process of making red blood cells is called erythropoiesis. And this is where we have basically the production of red blood cells in bone marrow. So in adults, this is called myeloid tissue, red bone marrow. In children, or I should say in fetal and embryonic development, what we find is that the bones are not yet fully formed. They don't have the bone marrow cavity completely formed. So they don't have the mature myeloid tissue like the adult does. So what they have is they've got a little bit different means of making red blood cells. So they basically have embryonic blood cells moving from the bloodstream to the liver, spleen, thymus, bone marrow to differentiate the stem cells. And these are going to divide to produce blood cells. So it's a little bit different from in the adult. So then we can look at something called hemocytoblasts, and these are also called hemopoietic stem cells. These are important for producing all of the formed elements. We have two types of hematopoietic stem cells. One are called myeloid stem cells, and this gives rise to all the formed elements except lymphocytes. And lymphocytes have their own stem cell called lymphoid stem cells. Everything else, all of our erythrocytes, our 4 of our 5 leukocytes, and our platelets are going to arise from myeloid stem cells. And really, the platelets themselves come off a giant cell called a megakaryocyte. And we'll talk a little bit about that later on. But all of those cell types are going to be derived from myeloid stem cells. Lymphoid stem cells are always going to give rise to lymphocytes. Now, when we look at red blood cell development, then we will see that there are several stages with which the red blood cell goes through in which it matures from what we call a proerythroblast to where it loses all of its organelles in its nucleus until it becomes a reticulocyte. And the reticulocyte is then ejected into circulation. And within a day of circulation, the reticulocyte becomes a mature red blood cell. So if we look at that process, we'll see that we've got in red bone marrow, here's a proerythroblast, and then it goes through these different types of erythroblast development stages. And we're not going to make you know these, but you will see that what's going on here is this is becoming less and less like a stem cell and more like a blood cell. So we're beginning to lose our organelles. We're going to eject our nucleus. A normoblast will eject its nucleus, become a reticulocyte. And the reticulocyte is going to be able to enter the bloodstream. And within 24 hours, it will have matured into this red blood cell, which will last typically about 128 days. We have a type of hormone made in the kidney called erythropoietin, and this is going to be secreted whenever there are low oxygen levels in peripheral tissues. So we'll see the kidney and the liver both will produce this erythropoietin. So this is very important, let's say, if you go up to altitude. So let's say you're climbing some mountains, and your mountain, let's say you want to climb Denali, which is a little over 20,000 feet. Well, you're going to have to acclimatize, and that is get the body used to lower and lower and lower concentrations of oxygen in the air, not because the components of the air is changing. We're not getting less oxygen concentration in the air. We're just having less air. So, in other words, the percentage of oxygen doesn't change in the air, but the air is rare. It's getting thinner as we go higher. And this is why aircraft that fly above 18,000 feet, or what we call the flight levels, have to be pressurized. So the cabins are pressurized. And even I believe it's mandated that you have to start pressurizing them at 14,000 feet, or you have to have another oxygen delivery system, either a mask or a cannula, because above this altitude, your body doesn't react well to the lack of oxygen. Now, you can climb mountains up to 20,000 feet, 22,000 feet without supplemental oxygen. There are people who have even climbed Everest without supplemental oxygen, but I wouldn't recommend it. In any case, whenever you starve your tissues of oxygen, you have a condition known as hypoxia. And in some of these super elite mountain climbers who have climbed Everest without oxygen, you look at their brains and they have these little lacunae in them, which are little chambers where there's nothing because some of that tissue got starved of oxygen and they have hypoxia. So typically when people go to high altitudes, if they go above 20,000 feet, then they will start using supplemental oxygen, or 22,000 is typically where people start using supplemental oxygen. People will go up to 18,000, 19,000, 20,000 feet all the time without supplemental oxygen. However, you can't do this in a short period of time. If someone were to be taken from sea level to 20,000 feet, they'd be passed out in minutes. So this is why it's so important to go through steps called acclimatization. So if you're going up to altitude, you have to take it in steps and take maybe 10 days to 2 weeks to get to high altitudes to give your body time to acclimatize. And that will allow you to produce more red blood cells, and it'll also allow other chemical changes in the blood to take place that allows for more efficient carrying of oxygen. So erythropoietin, or the hormone that produces more red blood cells, is one of the things that changes. The secretion of erythropoietin will become increased as you move to higher and higher altitudes. Now, one of the things that happens is something called blood doping, and you probably all heard of this. It's not a good idea at all, because what it does is it creates a lot more formed elements in blood suddenly. So that's going to increase the viscosity of blood as well. So what happens is some athletes, especially your distance athletes, it was especially prominent in Tour de France riders. So people who would do these long bicycle races, sometimes marathoners and so forth. But they would take their blood, they would withdraw the blood and freeze it and allow their body to make more red blood cells and get back to normal. And then they'd inject the blood that they had taken out earlier into their circulation. So they would have more red blood cells and supposedly more oxygen carrying capability. The other way that athletes cheated was they would take erythropoietin. So both of these were ways that athletes would use to increase their red blood cell counts, which turns out to be a very bad practice because it also increases the viscosity of blood, which reduces the flow, and, well, it makes the heart work harder because the heart has to pump harder against a liquid that is more viscous and has created more peripheral resistance in the blood vessels. So this is why it's a bad idea. Some people have fallen over of heart attacks due to this, or just their heart can't keep up with the increased peripheral resistance that this can cause, this blood doping. Another way that athletes have gotten around this, which works pretty well, is they'll go up to altitude and they'll train, and their body will naturally make more of this red blood cells, and then they'll come back down to wherever their race is being held, whatever altitude it is, and they will have more red blood cells. This is not considered blood doping, but it has its own risks involved. And I can tell you, coming down from high altitude, suddenly going to low altitude, it will create some problems. So you don't want to move very quickly from high to low altitude or low to high altitude. If you move too quickly from low altitude to high altitude, you can have things like cerebral edema and things like that. So you got to be very careful in any situation. You got to be careful about changing the amount of formed elements that you have at any given moment by large amounts, because it can result in all kinds of problems. As we've said, erythropoiesis is going to require B vitamins and iron. And obviously you have to have amino acids, because amino acids are what we make hemoglobin out of. Next, we'll talk about hemoglobin recycling.